Dichotomous key to different orders of insecta. Hind limbs always large and adapted for jumping. Pronotum large and produced downward at the side to form a collar behind the head. Forewings modified into tegmina. Auditory tympanum present. Orthoptera. Then hind limbs not always large and adapted for jumping. Pronotum not large and produced downwards at the sides to form a collar behind the head. Forewings not modified into tegmina. Auditory tympanum absent. 2. Mouthparts sucking and piercing type. 3. Mouthparts not sucking and piercing type. 4. Head usually horizontal with proboscis rising from the front. Forewing modified as hemiptera contain veins. Wings held flat over the abdomen at rest. Head usually vertical with proboscis rising from the back. Forewings either membranous or slightly thickened. Wings held roof-like at rest. Homoptera. Then forewings modified into chainless ornithopter, not filled for bright hind wings, folded crosswise and lengthwise and hidden under elytra. Antennae with 11 to 14 segments. Abdomen not slightly constricted at the base. Prothorax not fused with metathorax. Then pharythra not with variably modified spots. That is in case of Coleoptera. Then forewings smaller in size. Abdomen strongly constricted at the base. Prothorax fused with the mesothorax. Pharythra with variably modified spurs. Hymenoptera. First, dichotomous key to the families of order Hymenoptera. Basal abdomen segment modified into a true limb or node. Antennae always geniculate. Formicidae. Basal abdomen segment with or without some constriction, but always without true node. Antennae not always geniculate. Then body covered with branched or plumose hairs. 4 segments of hind tarsal broad, flattened and with hairy scrap apidae. Then body covered with simple unbranched hairs. Segments of hind thorax slender without such hairy scrap. And both often heavily sculptured with metallic colors. Last abdominal fragile apically notched or toothed, that is Crassididae. Body was sculptured without metallic colors. Last abdominal tergale not notched or toothed apically. 4. Then pronotum reach back to tegula and overlaps tail at an acute angle. Eye usually emarginate. Waspidae. Pronotum never reach back to tegula. Hind angle often produced into a collected tubercle. Eyes not usually emergent. Spacidae. Dichotomous key to the families of order Coleoptera. Head produced into a rostrum or beak with mouthparts at the tip. Antennae geniculate. 5 segmented with hairy parts. Bottom surface of about 3 segmented and 4 segmented extended by biological 3rd segment. Head not produced into rostrum. Antennae not geniculate. 3rd hairy ports and 4th segment not covered. Measurement of soil moisture. Aim to measure the moisture content of the soil. Procedure: 20 grams of fresh soil sample is taken and was properly weighed to know the initial weight of the sample soil. Then it was air dried on oven at a temperature 105 to 106 degrees Celsius for 24 hours. The soil is then cooled and weighed to know the dry weight of the soil sample. The soil moisture content is estimated from the fresh weight and dry weight of the soil. Moisture content, fresh weight minus dry weight. Moisture percentage is calculated from the value of moisture content by multiplying the moisture content by 100 into 100. Fresh weight of sample soil, dry weight of the sample soil, fresh weight minus dry weight by dry weight into 100. Result, moisture content of the fresh soil. We got the result. Then sample water content of the soil. Initial fresh soil 20, final 8.66 gram. Moisture content 11.34. Moisture content fresh weight minus dry weight 11.34 gram. Then moisture content of fresh soil in percentage. Initial value minus final value by 20 into 100. Then water holding capacity. Soil vary in different ways. They differ in texture, infiltration rates, permeability, depth, organic matter content, and water holding capacity. Soils are made up of three main parts: sand, silt, and clay. The proportion of each component determines the soil's texture. Each soil texture has its own water holding capacity. The amount of organic matter affects the water holding capacity to a degree. Soil vary in different ways. They differ in texture, infiltration rates, permeability, depth, organic matter content, and water holding capacity. Soils are made up of three main components: sand, clay. The proportion of each component determines the soil texture. Each soil texture has its own water holding capacity. The amount of organic matter affects the water holding capacity to a degree. Water holding capacity is the ability of a certain soil texture to physically hold water against the force of gravity. It does this by soil particles which hold water molecules by the force of cohesion. Due to the size of soil particles, the cohesive properties are much different. Procedure: take 20 gram of soil from the yard and homogenize it and dry it in the oven overnight. Take a funnel and line it with Wortmann 1 number filter paper. Place the funnel on a conical flask measuring cylinder. The dried sample is weighed and transferred to the funnel top. 15 ml of water is gently poured over the soil. Let the water drip in flask. The volume of water is measured by using a measuring jar, and the weight of the wet soil is also taken. Water holding capacity of the soil sample is in this much percentage. Calculation: water holding capacity WHC percentage. Water holding capacity percentage is equal to W1 minus WO by W2 minus W1 by W1 minus WO, where weight of the filter paper 0.60 gram, weight of the funnel WO 31.24 gram, weight of the funnel with the dried soil W1 31.24 plus 0.60 17.22. Weight of the filter paper plus weight of the funnel plus weight of the funnel with the dried soil. Weight of the funnel with wet soil. Then WHC is equal to W1 minus WO minus W2 minus W1 by W1 minus W2 into 100. Convergent evolution. Convergent evolution is the development of similar adaptive features independently by the distantly related animals. Convergence is used due to simultaneous in the walk of life. Evolution by light by insects, reptiles, and birds and mammals. They have all developed wings in response to the adaptation for living in the air. As a result, organisms with different body organization come to a pose of superficial resemblance. Convergence among closely related forms is called a parallel evolution. So, parallel evolution is the development of similar among types of adaptation among closely related animals by similar way of life. While convergence is the development of similar adaptation among distantly related forms, the convergent evolution leads to the formation of analogous similarities as they perform similar functions. So examples. The most common example of convergent evolution is the development of wings in insects, then flying reptiles, birds, and mammals, flying mammals. They have common features for the development of wings for flight. The aquatic vertebrates, cartilaginous and bony fishes, dolphins and porpoises are superficially alike, having fusiform or streamlined body with a median dorsal caudal and a pair of fins. In aquatic mammals and reptiles, the limbs are modified into slippers. The burrowing mammals, like moles, have them modified into slippers for climbing. Both two orders of class Mammalia. The order is Insectivorous, Lalatus, Ludens. Convergent evolution leads to the production of analogous similarities among the different groups or organisms, which indicate the evolution may lead to the superficial resemblance. Convergent evolution in wings: bat wing, bird wing, then insect wing. Convergent evolution in wing structure. Then forelimbs of vertebrates indicate convergent evolution. Okay. Then some other aspects related to this significance. The larger the region, the more worse the conditions, the greater the varieties of animals with results. Therefore, adaptive radiation is the evolution in several ancestral distribution straight from an uncommon and generalized ancestral type or one entity of an organism with the organismal stock. Mammals provide a classical example of adaptive radiation. The ancestry of all present-day type of mammals can be traced back to a primitive insect. Its 100 limbs were not marked for any particular type of locomotion. They lived on land and formed ancestors in motion. Mammals, which now have occupied 4 different habitats, have modification in their limb structure. May lead to arboreal modifications in squirrel and primates, lead to acquired modifications in animals adapted for flight, bats. One, cursorial modifications adapted to running forms, horse. The next line goes into fusorial burrowing mammals, echidna. The first line leads to aquatic conditions, whale, dolphin, etc. Then beak adaptations in finches, seeds, leaves, buds, fruits, insect, tool using finch. The similar divergent evolution has occurred in group of birds belonging to the family Fringillidae. Galapagos finches are now popularly called as Darwin finches. The mainland forms are all of one type, providing short, straight beaks for crushing seeds. From their central stock, they have undergone great diversification in their feeding habits and according to that in their shape and size of beak. Large ground finches, they have a typical finch-like beak which is correlated with the size of the seeds that they eat. And then cactus feeding. Large ground finches, cactus tree finches, they have a long, somewhat curved beak and for tongue for getting nectar out of. It also feeds on the soft pulp of cactus. And vegetation tree finches. This has a short, thick beak and short tongue for getting nectar from flowers. They also feeds upon the soft pulp of this. So insectivorous tree finches, they have a similar beaks as former when they are used for small. Then warbler finches, it has a warbler-like cylindrical beak which is used, and woodpecker finch, they have some other peculiarities. The causes for adaptive, once again different types are there we mentioned. Large ground finches. Large ground finches. Large ground finches. Cactus tree finches. Vegetation tree finches. Large ground finches. Large ground finches. Cactus tree finches. Large ground finches. Cactus tree finches. Vegetation tree finches. Insectivorous tree finches. Warbler finches and woodpecker finches. The cause for adaptive radiation are for food, safety and better fecundity ground. Naturally, the animals migrate to new habitats with little competition for better chances of survival. Adaptive radiation has resulted in the formation of new genes, subspecies, subclass and superclass. The macro and mega evolution may be arised due to phenomena of adaptive radiation. Sequidisc. Sequidisc created in 1855 by Petro-Angelo Secchi. It is a circular disc used to measure water transparency in oceans and lakes. The disc is maintained on a pole or line and then lowered slowly down into the water. The depth at which the pattern on the disc is no longer visible taken as a measure of the transparency of water. This measure is known as the Secchi disc depth. So this disc is maintained on a pole or line and then lowered slowly down into the water. The depth at which the pattern on the disc is no longer visible taken as a measure of the transparency of water. This measure is known as the Secchi disc depth and is related to the water turbidity. Various size of disc has been used since that time, but the most frequently used disc is an 8-inch diameter metallic disc painted in alternate black and white quadrants. The Secchi depth is reached when the reflectance equals intensity of light backscattered. The reflectance equals intensity of light backscattered from water. Secchi disc reading do not provide an exact measure of transparency, or there can be errors due to the sun's gaze on the water, or one person may see a disc at one depth, but another with better eyesight may see it at a greater depth. So, however, a Secchi disc is inexpensive and straightforward method of measuring water clarity. A Secchi disc measurement should always be taken off the shady side of the boat or cloak between 9 a.m. to 3 p.m. The period of best result is between 10 a.m. to 2 p.m. The same observation should be taken Secchi depth measurement in the same manner every time. Otherwise, fluctuations will be occurring. So, Secchi disc, we could see this picture. Alternate black and white color is there. So, a Bradley's funnel. A Bradley's funnel is a device which is used to extract insects from soil samples. It uses a heat source, a light lamp, to dry the sample, forcing the insects through a screen into a jar of preserving fluid. Aim to separate and identify soil arthropods using Bradley's funnel principle. Bradley's funnel work on the principle that insects and other arthropods are normally live in soil and litter will respond negatively to light, negative phototactic movement. Therefore, a light source is needed to force the arthropod to move downward, where they will fall into funnel and then into the collecting beaker containing alcohol. Materials required: glass funnel, heat source or bulb, large mesh screen, collecting beaker, alcohol for preserving, 70% ethanol. Then soil samples. Procedure: the soil sample was taken in a mesh and was placed on the top of funnel. Bottom of the funnel was directed towards the collecting beaker containing 70% ethanol. Allow the sample to dry slowly under the light. Sufficient distance was placed to prevent the funnel from overheating of the sample. The whole apparatus was kept undisturbed for 24 hours. Due to excess light on the top, negatively phototactic organisms migrate downwards and slips down into the collecting beaker. They are then preserved in the alcohol. Result: insects and other arthropods in soil are found to be negatively phototactic. And planktonic net. Planktonic nets. Planktons are floating and drifting plants and animals that live in ocean as well as in freshwater. They are most abundant in the ocean. Since most planktons are barely visible to naked eye, we must use a special net target to the small creatures to trap these small creatures. The plankton net is a funnel shaped fine meshed net that is put into the water. The planktons are collected by using plankton net from hundreds of gallons of water that pass through it. It is a cone shaped equipment used for collecting samples of plankton in stagnant bodies of water. It consists of towing line and bridles, nylon mesh net, and codent. It can be used for both vertical and horizontal sampling. Towing lines and bristles are upper part and it's used to hold the net. Nylon mesh is the middle part and is used to filter the plankton in the water sample in accordance with the size of mesh. Codent is located in lower part of the end of the funnel. It has a collecting cylinder and a value for opening and closing it. After that, it is pulled through the seawater. The do not pass through the net will be concentrated and trapped within the bottle. The first recorded use of plankton net was on John Boger Thompson. Then plankton net we could see how it is located. Iron rim handle, bolting silk net number 16, thick cloth band, plankton jar. Then plankton counting chamber. Plankton counting chamber, Sedgwick rafter counting chamber is used for the quantitative measurement of exact number of plankton in a precise volume of a fluid. It is constructed as a flat slide of dimension 76 mm into 40 mm on which it is cemented to form a chamber or a well on the middle. A chamber is 60 mm long into 200 mm wide into 1 mm deep. 60 into 220, 60 into 20 into 1. Okay. So, and its base is marked with a grid of 100 to 1 mm squares. This chamber is filled with liquid and a cover glass is placed over the chamber for examination and quantifying the planktons. Exactly 1 ml of volume of liquid can be dropped in the chamber. The plankton chamber is used in water analysis, culture, inspection, etc., where particles per unit volume must be determined. The cell can be used for quantification of living or preserved materials. While quantification, cover glass is placed over the plankton chamber. This to fill the cell, place the cover glass across the chamber top. This allows the air bubble to escape during the filling procedure. The sample is then taken into a 1 ml wide mouth pipette and then carefully transferred to the chamber. Do not overfill the chamber because the volume of the sample in this chamber must be known exactly and the cover glass must not float free but held onto the cell walls by surface tension. During counting, water may evaporate from the chamber. To prevent gas bubble formation, a small drop of distilled water may be placed on the slide along the cell, just touching the wall and cover glass. Before the cell count, Sedgwick rafter chamber should be allowed to stand for at least 15 minutes to allow algae or particles to set down to the bottom of the chamber. Plankton net counting chamber, Sedgwick rafter counting chamber, chamber wall. Lux meter. A lux meter is an equipment that measures brightness of falling on an object at a particular area. In other words, it properly gauges the intensity at which brightness appears to the human eye. Lux can be simply defined as a unit of measurement of illuminance or more accurately brightness. Lux meter for brightness. It derives its name from candela, which is standard unit of measurement of power of light. There are many benefits of using lux meter. Firstly, they provide quantified measurement of light available without which you are simply guessing. Secondly, you can clearly see and compare the impacts of different lighting conditions. It works by making use of a photocell to capture light. The lux meter then converts light into an electrical current stream, and other measuring this current allows the device to calculate the lux value of that light it has captured. Since light usually contains different colors, different wavelengths, and reading represents the combined effect of all of these wavelengths. Secondly, standard colors or color temperature are expressed in degrees of Kelvin. The standard color temperature for the calibration of most lux meter is 285 6K, an amount that is more yellow than pure white. lux meter, light receiver, screen, altimeter. After this we will be seeing all the subjects. Okay, so altimeter is an instrument used to measure the altitude of the object above a fixed level. Altimeter is an instrument used to measure the altitude of an object. You could guess that from the name altimeter, altitude. Object above a fixed level. The measurement of altitude is called altimetry. Altitude can be determined based on the measurement of atmospheric pressure. The greater the altitude, lower the pressure. The two main types are the pressure altimeter or solenoid barometer, which approximates altitude above sea level by measuring atmospheric pressures, and by radio altimeter, which measure absolute altitude. So distance above land or water based on the time required for a radioactive signals to travel from an airplane, a weather balloon or a spacecraft to the ground and back. Altimeter are important navigation instruments for aircraft and spacecraft pilots who monitor their height above the Earth's surface. Skydivers and mountaineers also use altimeter to pinpoint their location in the sky or on the ground. An altimeter reading change as elevation changes. The reading can also be changed due to weather as air pressure decreases during storms. There are different types of altimeter as a part of an area location, like barometric altimeter based on air pressure. GPS provide altitude as part of area location. Then radar, laser altimeter work similarly to sonar measurements on the sea floor. Types, which are the types? Barometric altimeter, GPS, radar and laser altimeter. Anemometer. An anemometer is an instrument that measures wind speed and wind pressure. Anemometers are important tools for meteorologists who study weather patterns. They are also important to the work of physicists who study the way air moves. They are also important to the work of physicians who study the way air moves. The most common type of anemometers has 3 or 4 cups attached to horizontal arms. These arms are attached to vertical rod. As the wind blows, the cup rotates, making the rod spin. The stronger the wind blows, the faster the rod spins. The anemometer counts the number of rotations or turns, which is used to calculate wind speed. Wind speed can also be determined. The wind pressure or speed. A tube anemometer measures the air pressure inside a glass tube with a closed one at one end. By comparing the air pressure inside the tube to the air pressure outside the tube, wind speed can be calculated. So anemometers are used at almost all weather situations, from the frigid Arctic to warm equatorial regions. Wind speed helps indicate a change in weather patterns, such as approaching storm, which is important for pilots, engineers and climatologists. So aerospace engineers and physicians often use laser anemometers. Anemometer, airflow, rotation, rotating magnet.